we can get you pulled up. Thank you very much.
Hi, everyone. My name is Tasfia. I'm a second-year medical student here at UT, and my project was determining significant polymorphisms in the choline metabolic pathway in BXD mice for the treatment of fetal alcohol spectrum disorders. So a little bit of background on fetal alcohol spectrum disorders. It's a group of conditions caused by being exposed to alcohol before birth. And while certain physical symptoms may present, every patient diagnosed with FASD has some form of cognitive deficit due to damage to the central nervous system. And here's just a graphic kind of identifying significant physical, cognitive, behavioral, and adaptive changes that may be impaired. These people are at a high risk for ADHD, mood disorders, depression, and dysregulation of sleep. So as you can imagine, can really impact quality of life. This is just another graphic kind of showing periods of gestation where the embryo is vulnerable to certain developmental defects. And as you can see, even as low as week 3, there's a vulnerability to central nervous system developmental defects. So that's pretty significant because a lot of women may not know that they're pregnant at 3 weeks, consume alcohol, have a child with these disorders. And so there's like a huge need for treatment, especially because the current treatment options are only symptomatic relief. So a little bit of background on choline. It's an essential nutrient and methyl donor involved in many physiological processes, including metabolism and the transport of lipids and methylation reactions and neurotransmitter synthesis. Key thing here is that when you're pregnant, you actually have an increased need for choline, and consuming alcohol also decreases your choline levels. So kind of you have this double whammy effect. If you're pregnant and consuming alcohol, you have a very significant decrease in choline and you're very deficient in the nutrient. And there were 2 studies done. Well, there were multiple studies done, but 2 that I highlighted here. When given as a supplement during pregnancy and early childhood, even to individuals not exposed to alcohol, choline was shown to improve memory, cognition, attention, and offered a beneficial effect to individuals with the listed disorders. And also in a double-blind randomized control trial performed comparing choline supplementation to placebo on 31 children 2 to 5 who had FASD. They found that children who received choline had a higher nonverbal intelligence, a higher visuospatial skill, higher working memory, and all these other benefits. So this led researchers at our lab to want to explore choline as a FASD treatment option further. So they explored whether the addition of choline to ethanol-exposed mice would decrease cell death in the forebrain and brainstem when compared to the control. And they did this by identifying 4 strains that were highly vulnerable to ethanol teratogenicity, which were BXT2, BXT51, BXT73, and B6. And then they took pregnant mice from those 4 strains and gave them either ethanol or isochloric maltodextrin, and then supplemented them with choline in one of 3 doses, 0 milligrams per kilogram, 100 milligrams per kilogram, and 250 milligrams per kilogram, and then performs tunnel staining to analyze cell death in the forebrain and brainstem. And what they found is, with the exception of the developing brainstem of BXT73 mice, choline reduced cell death in all the strains of ethanol-exposed mice. And the really significant thing here is that the level of cell death reduction varied based on the strain and dose. And here's kind of a visual representation of that. The key thing to kind of pay attention to is I'll draw your attention to the brainstem comparing A and B, so the B6 and BXT51 mice. At 0 milligrams per kilogram, an ethanol-exposed mice had a large amount of cell death in the brainstem. When given 100 milligrams per kilograms of choline, that cell death reduced significantly in both the B6 and BXT51. But then when given a 250 milligrams per kilogram dose, the B6 mice actually had a decreased benefit of choline, and BXT51 had an increased benefit of choline. So this led to my research project, which was kind of identifying what are the things that are causing this differential response to choline as treatment for FASD. So what I did was I kind of took the results from that research project and also explored these other studies. One that identified that 2 enzymes polymorphisms in two enzymes in the choline metabolic pathway accounted for significant differences in the level of choline found in the breast milk of mothers given the same quantity of choline supplementation. And also another study that found that polymorphisms in SLC44A1 caused significantly greater pre/post-intervention cognitive improvement when compared to those with other polymorphisms of the same gene. So my research question was, what are the possible genetic polymorphisms within the choline metabolic pathway that impact the efficacy of choline treatment for FASD? So first, what I did was I did a literature review and explored which genes are involved in the choline transport and metabolism in the liver, which is the primary site of choline metabolism, and I identified 22 genes. And then I determined whether the 4 mice strains from the previous study expressed the B6 or D2 genotype for each gene, and was able to rule out 10 genes with no variance between the strains, because we can kind of infer that if there's no differences in those genes between the strains, that probably wasn't the cause of the differential response. And then I did a liver expression analysis. So I identified if the liver expression of the genes varied based on the strains. So I again took those 4 strains, identified what the liver expression levels of the genes were, and if they were similar, ruled those genes out, because again, probably not the cause of the differential response. And then I did a liver expression versus percent cell death analysis based on the previous research results as well. So basically I compared the percent of cell death within the brainstem and the forebrain at the different dosages, and try to identify, and then identified an R-square value for all of the analyses. And here I have them written down based on the genes. And so we have the brainstem at 100 and 250, and then forebrain at 100, forebrain at 250. And then in light green, I highlighted all R-square values above 0.5. And then if any of the genes had 3 out of 4 or 4 out of 4 light green squares, I highlighted those genes in dark green squares, because those were likely the genes of interest. And that was my final gene list, kind of those 4 genes. So CHKA, CEPT1, well, this has more information on all the genes, but so CHKA, CEPT1, SLC44A1, and SLC44A3. And then I also did a human SNP. So I took the human version of these genes and explored SNPs, and I found that significant associations with alcohol use disorders, liver disorders, and developmental disorders for all 4 of the genes. And another thing that was really significant, I don't know if you guys remember, I mentioned earlier, but SLC44A1, there was already research done on polymorphisms with the gene, and they did find that differences in that gene accounted for differences in choline response and choline efficacy, so as a treatment for FASD. So future actions: test to uncover which SNPs most heavily impact efficacy of choline treatment for FASD symptoms, test different choline dosages as FASD treatment for each SNP to determine ideal choline dosages for differing genetic backgrounds, and then also expand to other organs in the choline metabolic pathway. And here are my acknowledgements and references.